Hi, my name is Lauren Gottschalk. I'm a master's student here at WKU. Today I'm going to be presenting to you an article on 6S RNA. The article was written in 2000 and it is entitled 6S RNA Regulates E. coli RNA Polymerase Activity. So we'll continue on and talk about the article. So here I have just a simple outline for the presentation. I'm going to give a brief introduction about the paper, the main points of the paper, then go into the hypothesis, methods, data and results, and finally a conclusion. So, to begin with a brief introduction, I just wanted to go over two things that I thought were important before I went any further into the article. So, first I wanted to discuss sRNA and 6S RNA. 6S RNA is actually a type of sRNA, so it's the biggest focus of the paper, so I wanted to address that. So sRNAs are small regulatory RNAs. They regulate gene expression during periods of stress. So 6S RNA in E. coli is actually the first sRNA to be sequenced. It was sequenced over 30 years ago, but until this paper, the function remained unclear. So the scientists of this article were really interested in uncovering the function and the role of 6S RNA actually with RNA polymerase. So I'm going to go over this in more detail and explain everything that the scientists found, but just to summarize that they found in their results, they did discover that the 6S RNA actually forms a complex with RNA polymerase and modulates sigma 70 holoenzyme activity. Okay, so this slide is just a super quick background, and I just wanted to just briefly go over this. What you're looking at here is the sequence of, well, it's the diagram of the 6S RNA. And so some previous research showed that the 6S RNA is transcribed as part of a dicistronic message that encodes for two genes. So in that message there is a gene that encodes for 6S RNA, and then there's an open reading frame for the gene YGFA. So at this time, they did not know the function or purpose of YGFA or 6S RNA actually at this point. So mainly, if you look here at the diagram, you'll see that the structure is pretty much double-stranded throughout the entire thing. Now, there are differences here. So we've got a few bubbles that I've underlined for you. They are just those small interruptions within the double-stranded. And then we've also got a huge central bubble or central region that is single-stranded. Okay, so here I'm just continuing a little bit on that background information, and I just wanted to take a step back here to talk briefly about bacterial metabolism in general. So during the growth, there are four stages. We've got the lag phase, the log phase or exponential phase, the stationary phase, and then the death or decline phase. So in the lag phase, the bacteria are kind of just becoming accustomed and adapted to their environment and the nutrients available there. In the exponential phase, that's when the growth's occurring, the rapid doubling, tripling, really quickly growing there. And then when we move into the stationary phase, this is where the nutrients are. Well, this is where the balance is coming in. So no further growth is continuing, and that will just go on until the nutrients are depleted, and the cell will move into the death or decline phase. So I just wanted to say really quickly, as some more background, in bacterial metabolism, there's something called catabolite repression, and so bacteria basically has a preference of its energy source, and usually that's glucose and usually lactose afterwards. And so basically, it's going to want to use the glucose first. That is its preferred energy source. So basically, if that's available, that's what it's going to use first, and then it will go sort of down the list of what else is available to the cell, and that's what it will metabolize. But the point of that is eventually it's going to run out of energy sources. So whether it wants to metabolize it or not, it's eventually just not going to have an option. Nothing else is going to be left. So at that point, it's going to go into the death or decline phase. So the reason I'm bringing this up is because one of the main focuses of this particular article is talking about the stationary phase, and it's discussing about how there are ways to overcome this nutrient depletion issue and things like that, of that nature. So I just wanted to explain that the cell will do what it can to a certain extent to metabolize whatever's available, but then at some point it's just not going to be able to do that anymore, and it's going to decline. So there's no getting away from that, but there are methods of stalling that, should I say. So I just wanted to quickly just bring that up, since that's going to be part of the discussion for this. For this slide, this is just a continuation of the background about bacterial metabolism. So I just want to say that in bacterial transcription, there's usually a -10 and -35 element in each promoter. Okay, and so the promoter is the biggest thing, because this is where RNA polymerase recognizes it and binds to it. It will bind to the promoter, and then once that RNA polymerase is bound, the DNA can move on, and transcription can initiate. So RNA polymerase is huge, of course. It's critical to transcription, and so are the promoters, because RNA has to be able to know where to bind and to know where to start in order for anything to occur. So the interesting thing about RNA polymerase is it actually has two forms. So there's the core form that's responsible for elongation and transcription, and then there's also the holoenzyme form, which is responsible for that promoter recognition. So the holoenzyme form is shown here. I've gotten that for you. And a holoenzyme, just really quickly, is a compound that's formed by a combination of an enzyme with a coenzyme. So here I've got a holoenzyme. This is actually the holoenzyme form of RNA polymerase, and as you can see, it's got 2 alpha units, 2 beta units, and then a sigma 70 area. I'm showing you this now because you can also see there's the 6S RNA, which is what I'm going to be discussing in the article. But I wanted to just say that this will make a lot more sense here shortly. The results of this experiment are that they did find that the 6S RNA binds to this part of the holoenzyme complex and forms this. So it's going to make sense here shortly, but if you're wondering, like, why is that there already? Isn't that what we're trying to find out? Yes, that's what we're trying to find out. So I will, it will make more sense in just a few minutes. Okay, so this is figure 1. This is where I'm going to start talking about the article itself. Before I go into this further, I just wanted to apologize because I went over this presentation and explained everything with the pointer, and then I was having technical difficulties and could not get my audio to upload with it. I don't know what happened. But anyway, so my marking's already on here. I apologize, but I will do my best to direct you on what I'm discussing. So, so first, the researchers wanted to see what 6S RNA interacted with. So they knew it was important. They knew that it had some sort of role with RNA polymerase, but it had been 30 years and no one found anything. So this was a starting point. So that previous research did show that 6S extract migrates at 11S and deproteinized 6S migrates at 6S. So they used something called gradient fractionation, which is a method in which you centrifuge a sample and then a fractionator cuts it into fragments for individual analysis. So they used that, and they used E. coli K12 cells. They fractionated them, and the total RNA was isolated and analyzed by northern and western hybridization. So here I have figure 1, which is showing those results. If we're looking at part A, so we're looking at, so we're looking at lanes 11 through 14 in the top box. Okay, that dark banding, really dark bandings, are suggesting an abundance of 6S because we know that the intensity of that darkness tells us roughly how much is present. So you can also see a little further down, I've underlined an area from roughly 19 to 20. And so that's kind of another peak there. We can see an abundance there. It's just not quite as strong as the first abundance. The bottom box, so we're still on part A, we're in the bottom box, shows the Western analysis using an anti-serum that recognizes RNA polymerase subunits. So I underlined on the right-hand side the subunits. We've got the alpha subunits and the beta and beta prime subunits. So we notice the similar banding in part A, roughly, if you use the numbers, I know they're not roughly in the same area. So just say the first piece that I underlined and then the second piece that I underlined. We're seeing a similar pattern. We're seeing a huge dark banding in the center there, showing that abundance, and then just a few areas down, we're seeing another peak, just not quite as strong as the first one. So these similarities kind of provide evidence that 6S co-fractionates with these subunits of that RNA polymerase complex. So this is the first kind of, okay, hey, we have evidence that there is an interaction here. Moving on to part B, we're comparing the weights of the 6S RNA and the subunits of RNA polymerase. So on the right-hand side, I've underlined the beta, beta prime, and alpha subunits. And then I've also semi-circled, squared, boxed in, whatever, the dark banding there, and it's roughly 40-ish kilodalton in size, which was later identified as the alpha subunit of RNA polymerase. So the researchers did a bit of outside research as well, and they didn't document the results like with graphs or anything, but they did mention in the report that this was further findings. Okay, so moving on, the researchers wanted to know, okay, is there like what's going on between 6S RNA and RNA polymerase? So they want to know if there's a physical binding or attraction. So to analyze this, they used something called immunoprecipitation, which is a method of precipitating an antigen for analysis using antibodies. So they used K12 cells and antibodies that recognize the subunits of RNA polymerase. Okay, so figure 2 is showing the results of this. In part A, we're looking at part A, and let's see where I want to draw your attention. So we're going to start, so there's the 3 boxes of part A, okay, the shaded boxes. So we're going to start in the first one, and we can see here, let's see, I'm trying to think of how I want to do this. Okay, so we can see that RNA co-precipitates with RNA polymerase antibodies, but not with the others. So that's the first 2 boxes that I've sort of semi-circled there in red. That's showing that there's nothing there, except for right there in the middle in that dark banding. So also when RNA polymerase is diminished from the supernatant, so now we're moving over to the right hand side, still in part A, but we're on the supernatant side, we see that 6S RNA begins declining. Okay, so I did not circle this part though. When we move though down 2 boxes to where we're looking at 6S RNA and 5S RNA, I did circle a little bit here. So 5S RNA, we can see that none of it was immunoprecipitated. So that's that first squiggly circled area. But we do see a presence in the supernatants, which I've underlined. Okay, this tells us that 5S does not bind to RNA polymerase subunits. Moving on to part B, the scientists wanted to see if they could figure out if 6S RNA binds to the core RNA polymerase or the holoenzyme RNA polymerase. So remember, I was telling you there are 2 different types, and we're trying to figure out which one it would bind to. So they did another set of co-immunosuppression, this time selecting for the sigma 70 subunit. So if we got a positive result, it would suggest the holoenzyme, because that's specific to the holoenzyme. If not, then it would be the core RNA polymerase. So I have circled that one little area of dark banding around the 6S that is in the anti-core region. So that tells us right there that this is the holoenzyme RNA polymerase that we're dealing with. Okay, so moving right along, so now the researchers have identified there's a interaction between the holoenzyme RNA polymerase, not the core. So now they want to see if that 6S RNA is present in purified RNA polymerase. So in figure 3A, we've got the predictive secondary structure of 6S RNA, which I've already shared with you. And then in B, we have the same thing, but we have inserted H. influenzae, which is HS RNA-like. So the scientists decided to target that region with an antisense oligonucleotide. I'm going to refer to that as HI6S. A bit of further data that was shown, remember I said that they did some outside research as well, but they did discuss it. It led them to the conclusion that the H influenza 6S RNA is a homolog of E. coli 6S RNA. So knowing that there's a possibility of other proteins being at play, the scientists use antisense selection with the oligonucleotide to identify other proteins. So looking at figure 3C, I did not circle this, but we have a band present at the beta and sigma 70 RNA polymerase subunits with HI6S, which shows us that the 6S RNA polymerase complex can be selected through the RNA. Okay, in figure 3D, we're comparing the sigma S and the sigma 70 factors. So the very dark line at sigma 70, which I have underlined there, confirms that 6S interacts with sigma 70 RNA polymerase holoenzyme specifically. To determine which RNA polymerase subunit directly binds to 6S, the researchers used cross-linking with radiolabeling of RNA. So that's shown in figure 3E. I did underline on lane 4, we show some banding for the HI6S RNA. And then, again, additional testing was done that wasn't presented, but revealed that that large band was sometimes there when 6S RNA wasn't there, but always enhanced when it was there. The small band in lane 2, which I underlined, was always there only in the presence of 6S RNA. The large band is presumed to be the beta prime subunits, and the smaller would be that sigma 70 factor. So using core and sigma 70 antibodies, the researchers completed a second analysis. And I put that's the second part of diagram E. So in this case, the samples were denatured so that the interaction was concrete, no room for speculation, no other variables present. So in lane 6, I did underline lane 6, we see that the larger band, and then lane 7, we see the smaller band, right at sigma 70, confirms that sigma 70 is where 6S RNA interacts. So next, the scientists wanted to see, okay, what point specifically is the 6S RNA interacting? So the levels of 6S RNA and 5S RNA were quantified through the different growth stages and analyzed by northern hybridization. In figure 4A, I drew a line, and even though it is almost linear, I realize that's misleading because it's not meant to be linear. It is showing the exponential growth of 6S RNA. So you can see there's a huge jump between that 0 and 24-hour mark. Part B is just confirming that in a different way. So we're looking at the northern analysis here. We have 6S RNA and 5S RNA. So what's interesting here is if you look at the purple underlines, both of them are really not, there's trace amounts. Okay, there's even less amount of 6S RNA than 5S RNA, but still, they're both pretty trace amount at this point between 0 to roughly 3 hours, which is fine. But what changes is that as time goes on and we get into the stationary phase, the 5S RNA does not change whatsoever. However, the 6S RNA, which I did circle it, accumulates much, much quicker and in a much higher abundance. So even at the 24-hour mark, it is at the highest level that it has been thus far. So that's interesting, and that shows us that 6S RNA is accumulating during that stationary phase, which I discussed earlier. We wouldn't really expect that normally, considering that stationary phase. We're not expecting a lot of accumulation or growth in that phase. So moving on now, the researchers, so now we know that there's binding to the holoenzyme complex. We know that this is happening in the stationary phase. We know that sigma 70 is where the 6S RNA interacts. So anyway, now the researchers are looking at that complex, the 6S RNA to RNA polymerase complex, and they're using wild-type K12 cells. So in figure 5, we're seeing that increasing amounts of RNA polymerase are appearing in the stationary phase. So if you look in between the boxes, so we've got the K12 and then the SSR1. If you look between them, you'll see 2 hours, 6 hours, 24 hours. Okay, the stationary phase is at 24-hour mark. So by 24 hours, comparing the two, it's basically just that 6S RNA that we're seeing in the K12 cells, which kind of confirms what we just saw in our last figure, that it's accumulating very highly, and that it's pretty much by itself in that point of the stationary phase. But then in the SSR1 cells, RNA polymerase is present in all the phases, but it's not shifting to 6S RNA like it is in the K12 cells. So in part B, we see no sigma. We're going down to part B. So in part B, we don't see any sigma 70 subunit in the SSR1 cell. So that's the green circle. You can see there's nothing there. But in the wild type, which is the yellow circle, we see that dark banding again, that huge increase in the stationary phase compared to that exponential growth that we would normally expect under other circumstances. So these results kind of hone in that interaction between sigma 70 and RNA polymerase is only present during high levels of 6S RNA in the stationary phase. Next, the researchers wanted to see if that 6S RNA alters the utilization of sigma 70 holoenzyme compared to the stationary phase specific sigma S holoenzyme. So they analyzed transcription using this RSD gene, using an RNase protection assay. In part A here, this is just a simple schematic of the RSD gene, really simplified, not really showing you much. The point of that is to show the two promoters that are present during the stationary phase. So you've got P1, which requires sigma S, and then you've got P2, which requires sigma 70. So moving on to part B, this is showing, so if you see we've got the K12 at the very top there, and it's showing some banding between both P1 and P2. So that's the yellow little lines there have shown, so that we're getting a positive result with both promoters and K12 cells. So, but in lane 2, here's where it gets interesting. So the SSR1, we don't see any change with the P1. They're the same intensity, or roughly very similar. But for P2, the banding intensifies a lot. So this kind of suggests that sigma 70 holoenzyme is more active, and it suggests that 6S RNA could alter the utilization of P2 relative to P1 in that RSD gene. So this is just giving us more fuel for understanding how 6S RNA specifically picks sigma 70 in that RNA polymerase. So lastly, we move on to figure 7. The researchers wanted to see if a phenotype for the absence of 6S RNA could be found in strains lacking sigma S. So they tested this by creating RPOS, which has mutants carrying the SSR1 mutation. So looking at the bottom here, you can see we've got the control, which is PKK asterisk. And then we've got 6 sRNA overexpression vectors. So that's the PKK asterisk 6S and the PKK asterisk 6S plus Y. So these were all grown, diluted, and then left to grow on agar. Figure 7 is showing the results here, and we can see there's decrease. Okay, we see that there's decrease, and even the controls. Well, in both, from the 12 hours to the 24 hours, we definitely see that decrease, but there's a much larger difference over the 24 hours. We don't see as substantial of a decrease in the 12 hours. So this is suggestive that wild-type 6S overexpressions could lead to reduced viability in sigma S deficient cells during stationary phase, which we kind of knew that because sigma S is huge in regulating growth during the stationary phase and aiding the cell in starvation situations. So that's it, guys. These are the results of the experiment and the study. So number 1, 6S RNA interacts with sigma 70 RNA polymerase holoenzyme. So this was shown to be highly specific and efficient. 6S RNA is the only RNA the only RNA found to stably bind to the RNA polymerase. Okay, number 2, 6S RNA accumulates in stationary phase and mediates growth phase specific alterations in RNA polymerase. So this is really important for cases of starvation in the cell. Anything that could possibly stall, the cell dying essentially, is going to be huge. So number 3, stable association of sigma 70 with core enzyme extracts is only observed in the presence of high levels of 6S RNA. So this just gives us more fuel to understand the importance of 6S RNA and its association with sigma 70 and that holoenzyme complex. And then lastly, 6S RNA modulates sigma 70 promoter usage in stationary phase. Okay, so I just wanted to say really quickly that in the discussion, the scientists suspected that, you wonder, okay, why all of this was found in this one experiment? Why wasn't anything else found in the 30 years since they discovered the sequence of this? And so they kind of think it's because all the other experiments use purified RNA polymerase. And they made sure to steer away from that to, test that and make sure that wasn't a variable here. And so they think that would be the reason. And so it kind of made them wonder if there were other, molecules or proteins that were falsely experimented on in the same way. So anyway, I thought that was interesting. So I just wanted to conclude this by talking about some future potential uses of 6S-RNA and the information found in this article. I just wanted to reiterate this article was published in 2000, so it's been 22 years. So of course there's been other things that have made headway, but just based on this article, the scientists identified a few things that could be looked further into down the road. So the first one is that 6S RNA and the RNA polymerase complex may contribute to altered promoter utilization during the stationary phase. So it could alter the promoter recognition and could decrease in sigma 70 dependent transcription. So this is huge, of course, because anything that's going to alter transcription is something that could be an area of research. The stationary phase is very important because of cell starvation in times of stress. So anything that could affect that, or the promoter recognition to affect transcription, that's always going to be interesting and something that could help with anything related to this down the road. The next thing was that the 6S RNA polymerase complex could be a mechanism for storing sigma 70 holoenzyme during starvation. So once again, this is huge. Even thinking as far as this could be an issue, antibiotic resistance. Understanding how starvation and cell death could be stalled or pushed back or even overcome, in certain ways is going to be huge for, targets of medicine and everything that we know about, this could be completely changed. So, that's huge. And then the last thing is 6S RNA may affect RNA polymerase release from DNA and possibly have sigma 70 reassociation with the core enzyme. And they said this because more RNA polymerase remains bound to DNA in the absence of 6S RNA. So, this is going back to the core enzyme. Personally, I think all the evidence they presented was really, really solid for just the holoenzyme, but who knows? Maybe the core enzyme is important in its own little way for this, just something that we don't understand, didn't understand at this point of the article. So, anyway, I enjoyed the article. I hope you guys enjoyed my presentation. If you have any questions, please comment them on the bottom of this post in the discussion board, and that's it. Thank you for your attention.